Yeah, good afternoon, class. I welcome you again to our another new segment today, which we are going to talk about protein synthesis or translation. I have came with some diagrams and information that we are going to use as a reference in terms of understanding of this process of protein synthesis, which we can also say translation. Just to make you aware that is one of the key areas which we just need to understand. By definition, if you see protein synthesis is a process by which proteins are manufactured. I'm sure that you understand when we say synthesis is manufacturing of proteins. So in this case, we are going to use our diagrams there that are just shown on the board, and then we would have the final explanation and definition of protein synthesis, as well as the explanation on how protein synthesis takes place. So I'm just going to quickly answer these questions. They are going to make us understand it better. Without wasting any time, we are gonna look at these questions. This is a question paper of 2023 November and December 2023. I mean, this November and June 2023. So I'm just going to show you and explain to you how this process is taking place. So let's quickly just answer the questions. We have got our diagram there that is showing a DNA molecule, and then we have got an arrow there that is showing our DNA molecule, which is shown here. You can see this is DNA, and then you can see DNA is giving a process, or it's forming mRNA that is shown through process Z there, which is transcription. But for in this case, let's just answer the questions, and then we'll answer all other questions and additionals after the discussion. So what we are saying is that the first question is saying 1.5.1. The first question is saying identify process Z. You see the process whereby DNA forms RNA, forms mRNA. We call that it's called what? Transcription. So process 1.5.1a, we are saying this is called transcription. Transcription is a process through which mRNA is formed from DNA, where a small piece of DNA unwinds, and then it forms two of its strands, or it gives a strand that is going to be used as a template in the formation of mRNA, which is going to be done through the pairing of the bases, complementary bases, which are in the nucleoplasm there. But in this case, we are not asked to explain the process of transcription, but DNA forms mRNA. That process is called transcription, which is shown there by process Z, that is indicating that is transcription. Molecule R, it is where, I mean, that is what? That is mRNA. That is 1.5.1b. We are saying this process, molecule R, it is called mRNA. mRNA is always formed when DNA forms, I mean, whenever DNA unwinds, and the process through which transcription goes through, when transcription is taking place, it forms mRNA, which is a single strand that is being highlighted here to say process, I mean, molecule R is the same thing that is shown through this broken arrow here, that is going to point to a single strand again that is coming out of the nuclear plasm here, or which is coming out of the nuclear membrane through the nuclear pore that is shown there through that broken line on our diagram. So the next question C is saying organelle Q. What you are saying is organelle Q, where mRNA goes through such that the mRNA codons are red, we call that it's called what? It's called the ribosome. It's called the ribosome. In other words, it is the manufacturing site of proteins through the reading of the mRNA, I mean the mRNA codon. This whole thing, which is being shown here, we are saying it is what? mRNA. So it will have to pass through, the what? The ribosomes that will identify the order of the codons there, so that a corresponding anticodon can be brought up through the tRNA. So this is mRNA that is being shown here, and then it passes through the ribosome, that is C there, organic Q. And then 1.5.2, the question says give the collective name for the nitrogen bases on O. We are talking about these nitrogen bases here. The nitrogen bases that are at O, we are referring to these nitrogen bases that are here. Their collective name, what are they called? We are saying these are called anticodons. They are called anticodons, these ones. They are called anticodons. Anticodons, which we are saying 3 nucleotide bases that are found on what? On tRNA. These are called anticodons. In other words, we are saying 3 nucleotides bases that are found on tRNA. You can see this is a tRNA, this one. This is tRNA. So the tRNA, this one, we are saying this is our tRNA. So tRNA contains the anticodons. tRNA always have anticodons. So the anticodons, they are the corresponding complementary bases that are found on the tRNA, which would have to bring the corresponding amino acids to what? To the ribosome there, in order to make sure that there is the protein synthesis that we are talking about. So 1.5.2, it is the anticodons. The answer just wants you to say anticodons, but for clarity's sake, now I just managed to give, or I've just said, these are called anticodons. The answer wants you just to say anticodons, but for the clarity's sake, now I've just defined what are those anticodons in this regard. So yeah, they are anticodons, 3 nucleotide bases that are found on tRNA. That's what we call anticodons. So the next question is saying, determine the sequence of nitrogen bases at area S. Determine the sequence of nitrogen bases at area S. Our area S, which is given here, is that we want the nitrogen bases that are found, the nitrogen bases that are found on area S. We are talking about this area S is this one. We are saying our nitrogen bases that are found here. So what is happening is that this answer number 1.5.3. 1.5.3, what you just need to understand here is that nitrogen bases on DNA, when we are looking at a DNA molecule, there is a complementary basis of nitrogen bases. So as we can see, in this case, we have got T here. So our complementary base that must be found here is that we have got T here. So what is happening is that the opposite of T, or the complementary base of T, is supposed to be what? It is A, which is on this side. And then we have got C here. The complementary base for C is what? Is G. And then we have got A there. The complementary base for A is T. So what we are saying is that these bases that are, or the complementary nitrogen bases on area S, it is AGT. GT is the complementary bases that are what? That are on area S, which is adenine, guanine, thymine. We are saying this is caused by the concept or by the fact that on strand 1 there we have got T. On strand 1 we have got T, we have got C, we have got what? We have got T, we have got C, we have got A. So here we are just giving the complementary basis of this strand that is given, the strand 1. So in that case, we are saying C pairs with what? C it pairs with what? With A. I mean T pairs with A, G pairs with, I mean C pairs with what? With the G, and then A pairs with the T, which is the reason why we are saying this answer here is GAGT. That is why we are saying our answer is what? AGT. So AGT is the order of the nitrogen bases that are found, the nitrogen base sequence that is found on what? That is found on area S. I'm saying this is the complementary pairing of the bases that is taking place at area S. This is the complementary pairing of the bases that is taking place at area S, which is determined by TCA that we have given here to say we have got TCA here. So its complementary basis is supposed to be what? AGT. AGT is what is the complementary basis of this case. So that is how the answer is calculated for that, to say we have got TCA, and then TCA there is the one that is defining the order of what? AGT that we have given there. So it's AGT. AGT is our answer in that case at 1.5.3. Question 1.5.4. Our question 1.5.4 is saying which strand 1 or 2 was used as a template for the formation of molecule R. Like we said, 1.5.4, we said that DNA is used, one of the strands of DNA is used as a template in the formation or in the formation of what? Of mRNA. So the question is saying which strand 1 or 2 was used as a template for the formation of for the formation of molecule R. In this regard, what we need to understand, class, is that we just have to calculate the order of the nitrogen bases that are on area R. So therefore, we can know that which one is the complementary pairing, which one is got the complementary basis for it. So R, we have got the nitrogen basis AC, AUC. This R, we are saying R is this one, which is the mRNA. R is referring to what? To the mRNA that is given there. The mRNA that I've ticked there is the one that is being given as what? Is our R in that case. That is our R, our mRNA. So the question wants you to understand, to say which strand. So in that case, what we just need to understand, we just, like I said, we look on the order of the nitrogen bases. So it is AUC. So we say we have got strand 1, we have got strand 1 here, and we have got strand 2 there. But which one was used as in the formation of this one? AUC. So we are saying the complementary bases that are found there. A must pair with what? With the T. So we have got T on strand 1, and then we have got A complementarily pairing with U, and then we have got G pairing with what? With the C. So what I'm saying is that our strand should be strand 1. It is strand 1. We can just say strand 1 there. The reason strand 1 was used, I'm saying, get it right, that we are saying you consider the order of the nitrogen bases that are found on mRNA versus the DNA. So like I'm saying, our DNA molecule, we can just give to say, let me just give to say that we have got our DNA molecule, which is here as the strand. We are saying the strand that was used, this is a DNA strand, this one. we can say this is our DNA strand. Let me just put it horizontal, so therefore we can just get it right. We are saying our strand of DNA can be that one. We can say our DNA strand there, our DNA strand, that is our DNA strand, which is supposed to have what? We have got, which we are saying here, we have got the nitrogen complementary bases there, which we have got T, we have got A, we have got what? We have got G. So the complementary, which is supposed to be forming the mRNA, we are saying which is supposed to form the mRNA there, our mRNA strand, we can say our mRNA strand is supposed to be what? Our mRNA strand. Or mRNA strand, it must have the complementary bases that must pair here. So we are saying we have got what? It must be mRNA strand, so we should have what? We should have A here, A pairs with what? With this with T, and then because it's RNA, we no longer have T, we have got U here, and then we have got what? We have got U there, and then we have got C here as the complementary base that's supposed to pair with what? To that one of G. G pairs with A, that pairs with that. So we have got A, UC pairs to what? To A, to TAG. That is given there. That is what is happening. And then just for clarity's sake, you can see again here at the center, we can see again here at the center of our R strand or our mRNA, we have got GGU. So GGU is given here to say that here at the center we have got we have got GGU. So GGU, obviously, in the complementary bases that must be found opposite to it, there must be what? We must have C, we must have what? Our CC what? Our CC, we are saying our complementary base must be our CC what? Our CCA. Our CCA, just to see that this is the way the orders, the complementary pairing must take place. So that is why we said what? That is why we said it is one. It is strand one. One is the one that is forming the strand that was used. Strand one is the one that was used to form what? To form mRNA. This is our strand one here. Our strand one. That was the one, or that is the one that you are seeing on here to say this is our strand one, which is what? Which is actually used in the formation of what? In the formation of mRNA. Strand one is the one that is used, is the one that is used there. Strand one is used. As you can see, the strand one is used. Okay. Question 5 point, next question. Our next question wants us to say, next question is saying 1.5.5, which amino acid 3, 4, or 5 will be brought to area P? Which amino acid will be brought to area P? Which amino acid will be brought? Next question, 1.5, which amino acid, which amino acid one or two was used? The next question we are saying, question number 5, 1.1.5, 1.5.5, 1.5.5 is saying which amino acid 3, 4, or 5 will be brought to area P. Our area P is this one. Our area P is this one. So which amino acid must be brought here before we even look on the order that is given there to say which one can be brought to area P? Which amino acid can be brought to area P here, to area P, this one. We are saying, how do we know that? In order for us to know that, we are saying area P is supposed to be just the it must be the completion of this mRNA strand, which you are seeing that we said that mRNA, this strand was formed through, this mRNA molecule was formed through strand 1. So we are having our first order of nitrogen bases, which is, we have got on this one AUC, which is here AUC. AUC is the one that is given here. We are saying our AUC is given. We have got also GGU. This is GGU, which is given also here, to say GGU is given there. Again, so therefore we are saying the last piece is this one, which is what? Which is asked to be on area, or to be on area P here. So which one must come here? It can be, is it 3, 5, or 4? So how do we know that? We just need to complete this strand, which we are saying it must be what? Having to say that we are going to use our nitrogen bases on the DNA molecule, which is our DNA molecule. We are going to use strand 1. Strand 1 is this one. Strand 1 is that one that I've marked. So we are saying we are given TC strand 1, excuse, strand 1, we are saying we are given our strand 1, this one, is to say we have got AGT. So the complementary basis that must be found there on this one is supposed to be, I'm saying it's supposed to be what? What is the nitrogen basis that must complete P? P, we are saying it must be having the nitrogen basis what? Molecule 1 is this one. So what is the nitrogen basis that must be found on here? We are having AGT. So our complementary basis must be what? It must be here. We must have U, because it's what is it has U there. We have got C. We have got C there, and then we have got what? We have got A there. So what we are saying is that our complementary basis that must be found on area P must be, it must be what? It must be UCA. It must be UCA. This must be UCA. We are saying to motivate our answer for you to understand, to say, why do we say that? What is, which, what is this? We are saying, what is it? The question is saying UCA, AGU. Okay, we are saying which one must be brought there. Therefore, we are saying our answer must be what? Our answer in this regard, we are saying it must be AGT, excuse, it must be AGU, which is like in this case here, our order of nitrogen bases that this person has been asking, I am sure that it was misplaced, because we should have UCA. UCA should be the correct answer in this case, which is in this regard, I see that is not given. UCA must be the order, because we've got A, G, T this side, which must complete this last part here, which we are saying it must be UCA, which is not given. UCA must be the answer in this case. UCA must be given. UCA must complete, or must complete, UCA must complete or be brought to what? To P, to area P. UCA must complete or be brought to area P there, which we can see from our answer, that from our options that are given there. We have got CCA, which is not there, which cannot be correct. We have got UAG, which is not there. We have got AGU, which is also not there. So the correct answer must be UCA. But in our case here, there is none of them which is given. Hence, we are saying it must be UCA, which is not given there. There is no answer, or neither of these ones that are given there can complete that. It cannot be 3, it cannot be 4, it cannot be 5, because the answer must be UCA, as determined by the order of the nitrogen bases that are found on what? On strand 1, this one. Thank you. So question last 1.5.6. 1.5.6, name the type of sugar that forms part of the structure of molecule R. Molecule R is this one. The sugar that forms that one is what? 1.5.6. The question wants you just to know, to say, what is the sugar that forms mRNA or RNA sugar? 1.5.6. The sugar for RNA is called what? Ribose sugar. It's called ribose, which we are saying sugar for RNA. The question wants you to know. So the answer is what? It's ribose. It's ribose sugar. That is what the question wants you to know. The question wants you to know what type of sugar is found on that strand R, or which is found on strand R. Strand R, it is what? Our strand R, we are saying it is ribose sugar. It is ribose sugar there. Ribose sugar is the answer in this case. Ribose sugar is what you need to understand. So it is done. Question number, first question. So that is basically what we are saying with regard to the questions that are given here. Identify the molecules that can be formed or that are happening here. We have answered the questions there, and we have given the motivations for those answers. Why are we saying that that is the process? Let's quickly just answer our next question. Let's just quickly answer our next question. Our next question is asking us, it wants us to quickly just understand what is happening here. Our next question that is given is also the same thing. I have just brought these questions to you so then you can be able to understand it more clearer than what we have done there. So here we are given to say this is a diagram that represents, it's a diagram representing the process that occurs during protein synthesis. So let us quickly answer this question for the interest of time. What are these? So then we can comprehensively cover the protein synthesis process. Identify molecules. Molecule X. Identify molecule X. Molecule X, it is an amino acid that is being attached to what? To tRNA. So we are saying this 2.1.1, it is what? The molecule XA. We are saying question A, 2.1.1a. We are saying molecule X is amino acid. It is the amino acid that is given, that is being shown there, is the amino acid. Amino acid is the one that you are seeing there. It's amino acid. Next question B. Question B is saying identify Z. Z, it wants you to understand to say what is Z there. Z is this structure, the single strand that we have talked about before again. What is Z? Z, which we said is a single strand. Z is referring to this whole strand there, which is shown here. Our Z is this one, which is having the nitrogen bases there, which is having the nitrogen bases. We have got C, A, U. We have got the middle strand, which is also given, which is AUG, AUG, and then we have got the last strand, which is also given there, which is G, A, C. So this whole thing is what is being referred to as what? This is our what? That is our Z, which we are saying Z is what? Is mRNA. It is mRNA again. Z is mRNA. And then give the nitrogen base sequence of the DNA base triplet complementary to the middle codon on Z. So I've already given you to say the middle codon that is being referred to in this regard is this one, where we have got ACU. I mean, we have got AUG. We have got AUG, which is given here. We have got AUG that is given there. So what is the corresponding, give the nitrogen base sequence of the DNA base triplet complementary to the middle codon. We are saying our middle codon that we are referring to, like I said, is this whole thing, which is AUG. So we are saying what is the complementary base triplets of the middle codon, which we are saying what are those one? What is it? What is the complementary of this one? It is supposed to be what? We are saying it is supposed to be A. There we have got T, which A pairs with T, as you can understand that. And then this is U pairs with A, and then we have got G here, G pairs with C. So we are saying this is the pairing that is being given in this case. This is the pairing that is being given. So our answer, which is question 2.1.2.1.2 A, 2.1.2a, we are saying it is supposed to be, the answer is TAC, according to the pairing of the bases that we have just talked about just now, to say A pairs with T, U pairs with A, G pairs with C. That's how this base pairing is going. It's TAC. The next question is saying, give the nitrogen base sequence of the nitrogen base sequence of what? Of Y. Y is this one. We are saying we are given the order of the nitrogen bases there, which is CAU. What is the complementary base pairing to these ones? What is the complementary base pairing to Y? So what we are saying is that C, it pairs with what? With the G, and then A. We are saying A, it pairs with what? Because this is what? This is RNA. So A, it pairs with what? With the U. And then we have got U here. U pairs with what? With A. So we are saying since it is RNA, T has been substituted by what? By U. Hence, our answer is our question B, 2.1.2b. 2.1.2b, we are saying what is Y? Y, we are saying it is what? GUA. GUA is our base pairing, is guanine, uracil, adenine. Guanine, uracil, adenine is the bases that are forming up or that are being complementarily pairing to Y, or which are forming Y there, that complementarily pairs to the CAU, which is given here. And then our next question is saying, number 2, name and describe the process shown in the diagram during the formation of protein. Name and describe the process. Name and describe, as you can see, the process that is being shown there is protein synthesis. Name and describe the process. So in this regard, you are supposed to understand the question is saying name and describe 2.13, 2.1.3. The process being shown is what is translation, which we said is the same as what? Protein synthesis. Translation is translation, which is the same as protein synthesis, translation. And then name and describe. Name and describe the process. We are saying each tRNA carries a specific what? Amino acid. As you can see, each tRNA, this is tRNA, it carries a specific amino acid that is shown there. Each tRNA carries a specific amino acid. In this case, for example, we are saying X there. It's an amino acid which is carried by what? By this one, which we are saying tRNA. This is tRNA. So tRNA carries a specific what? Amino acid that is being X there. So in this case, we are saying, let me just increase my spacing for us just to quickly just do this quickly. Name and describe the process. Name and describe the process. So we are saying first thing, each tRNA it carries a specific, amino acid. It carries a specific amino acid, that is the tRNA that I was showing there. It carries a specific amino acid. When the anticodon on the tRNA carries a specific amino acid, we are describing the process that is there. We are saying when the anticodon remember, we said the anticodons are the 3 nucleotide bases that are found on what? On tRNA. Each tRNA carries a specific amino acid. When the anticodon on tRNA on tRNA, like in this case, we will say we are saying a specific amino acid. For example, in this case, we are saying GUA. GUA matches the codon on mRNA. It has to match the codons on mRNA. It matches the codons which are on mRNA. In this case, we say GAU. It matches GUA, matches the codons on mRNA. In that case, we are saying in this case, which is I'm just giving the example to say what are the corresponding codons there, which is going to be CAU. I mean, CAU, we are saying this G pairs with what? With C, and then this one pairs with A, and then this one pairs with U. As you can see, GAU pairs with the specific codon CAU, which is given there, as you can see. So in that case, what happens is that GAU has to match with a specific codon, which is on mRNA, which we are saying GUA can pair with CAU, that you can see. TNR, in that case, what would happen is that our tRNA our tRNA, which we are saying that it will bring the tRNA will bring the specific tRNA will bring what? The specific amino acid that we've been saying will bring the specific required the required amino acid, which is, we are saying, according to what? The specific required amino acid bring the required specific to the what? As you can see, to the ribosome that is being shown here, to the ribosome, which we are saying why, to a specific required amino acid to the ribosome. The specific amino acid is brought by what? By the tRNA to the ribosome, as you can see there. What is next? What then happens is that the amino acid becomes attached to each other by peptide bonds. Amino acids the amino acid will become attached to each other each other. What happens is that the chain or these pieces of amino acids, they will become attached to each other by peptide bonds to form required proteins. They will become attached to each other, which we are saying, what is attaching them? The peptide bonds to form the required proteins to each other by peptides bonds to form the required proteins. So what we are basically saying, guys, is that proteins are actually formed as a result of amino acids that are joined together. In other words, we are just saying proteins, amino acids are the building blocks of proteins. When they join together, to form a series of amino acids. As you can see here, we are saying they join together and then they form a chain, which we're saying that chain is called a peptide bond, which is going to be formed, and then it will form the different it will form the type of the type of protein that is required. So in this case, what we are basically saying is that protein synthesis is the process through which proteins are formed, which is formed as a result of a combination of or a communication that is prompted by the ribosome as it reads the order of the codons on the mRNA. So if we can look at this, what we are saying is that we have got what are called codons and anticodons. So these, like we said, these ones are called anticodons. They are called anticodons, and then their corresponding amino acids or their corresponding nitrogen bases are called what? On mRNA, we call them codons. They are called codons. They're not the 3 nitrogen bases that are found what? On mRNA. So these are on tRNA, and these are found on what? On mRNA. mRNA, it gives what? Codons, or it has got codons, and then the tRNA has got what? It's got the anticodons. So as you can see, What we are saying is that that's the communication that basically takes place there in order for us to have the different types of amino acids or to have the amino acids being formed. It is as a result of the communication that takes place that happens to say that amino acids will become attached to each other by peptide bonds to form the required proteins as a result of the communication that is prompted by the ribosome as it reads through the order of the nitrogen or the order of the codons as they pass through the ribosome. That is what is happening. That is actually what happens there. So thank you very much for your time. Like I said, we have just repeated these questions just to make clarity to all of us that the question of protein synthesis, it is always given. It is always there, or it is mostly given whenever it is there. However, we need to understand how it comes, the format, the different formats through which it comes like. So I've given you this format B, and then I've given you again the previous format, which was format 1, which we said that this is format 1, and then we have also the other one, which was given as what? As format 2, which I have shown you there. Thank you very much for your time and all the best. Please don't forget to subscribe or follow our channel, Metric Hub, so therefore we can be able to continuously connect and see the next episodes of our lessons. Thank you very much and all the best. Good day.